Reabsorption of bicarbonate. Reabsorption of bicarbonate occurs through the nephron except in the descending limb of LH. In PCT, about 90% of the filtered bicarbonate is absorbed into the PCT. H plus secretion across the apical membrane of all of the cells occurs by both sodium-potassium antiporter and hydrogen ATPase. The sodium hydrogen antiporter is the predominant pathway for H plus secretion, and the uses the lumen to the cell, that is sodium gradient, to drive this process. Within the cell, the hydrogen and bicarbonate are produced in reaction catalyzed by carbonic anhydrase. The hydrogen is secreted into the tubular fluid, whereas bicarbonate exits the cell across the basolateral membrane and returns to the peritubular blood. The movement of bicarbonate out of the cell across the basolateral membrane is coupled to other ions. The majority of the bicarbonate exit via symporter that couples with the efflux of one sodium with one bicarbonate, that is sodium bicarbonate cotransporter. In addition, some bicarbonate may exit in exchange for chloride. Carbonic anhydrase is also present in the brush border of proximal tubule cells. This enzyme catalyzes the dehydration of bicarbonic acid in the luminal fluid and thereby facilitates reabsorption of bicarbonate ions. So this is how the reabsorption of the bicarbonates happens. So there will be active secretion of H plus ions is going to occur through the active sodium hydrogen pump, and this hydrogen H plus ion binds with a filtered bicarbonate to form bicarbonic acid, which forms water and carbon dioxide, and that is reabsorbed. And when it is reabsorbed, again in the cell it forms H plus ions and bicarbonate ions, and bicarbonates are transported to the capillaries, and that too it is occurs with the antipode with the chloride ions. In loop of Henle, the cellular mechanism responsible for reabsorption of bicarbonate in the thick segment of loop of Henle is very similar to that of proximal converter tubule. Potassium bicarbonate symporter in the basolateral membrane may also contribute to the exit of HCO3 from the cell here. In DCT and CT, intercalated cell secretes H plus ions and reabsorbs bicarbonate. Within this cell, the hydrogen and bicarbonate ions are produced by hydration of carbon dioxide, and this reaction is catalyzed by carbonic anhydrase. The H plus secreted into the tubular fluid via two mechanisms here. The first involves an apical membrane hydrogen ATPase, and the second, which couples the secretion of H plus with the reabsorption of potassium via hydrogen potassium ATPase, similar to that of found in the stomach. The HCO3, the bicarbonate, exists across the basolateral membrane in exchange for chloride, that is via chloride bicarbonate antiporter, and enters the peritubular capillary blood. This is how the bicarbonate reabsorption occurs in the late DCT. So one thing is, it is hydrogen potassium ion where it secretes H plus ions, and one potassium comes inside the cell, as a antiporter. The similar pumps, they are present in the HC gastric mucosa as well, and it can be secreted by active transport of H plus ions, H pump. So in a similar way, H plus ion is going to bind to bicarbonate, form carbonic acid, then water and carbon dioxide, which is reabsorbed, and in the cell again it gets converted back into H plus and HCO3, and HCO3 is antiported with the chloride ion.